So, first things, what is a eukaryotic cell? We know prokaryotic cells are cells that don't have membrane-bound organelles. Therefore, eukaryotic cells are cells that do have membrane-bound organelles. So, eukaryotic cells, these are cells that contain membrane-bound organelles. Okay, now the next question then becomes, okay, well, where did they come from? Were these two separate types of cells, that came up independently of each other? Or was there prokaryotes first and then there were eukaryotes? What happened with that? All of the evidence points towards the fact that prokaryotic cells were the very first types of cells, and then they evolved into eukaryotic cells. Okay, and they evolved into eukaryotic cells using two theories, or through the means of these two theories. And the two theories are non-conflicting theories. So a lot of times when people see theories, they're like, oh, one of them has to be right, one of them has to be wrong. Both of these are probably right. Okay, so the first one is called the Infolding Theory, which is sometimes referred to as the Theory of Invagination, which essentially translated just means infolding, right? And so the infolding theory says that cells will infold their plasma membranes in order to create compartments, and then those compartments became organelles. So the cell membrane infolded to create compartments, and then completed the infolding to create organelles. Okay, so we're going to go back to my example yesterday of the gym and the classroom, right? We talked about that in here? No? Yes, we did? Okay. Yeah. With the doors and getting in and out of the gym and getting out of the classroom, right? Okay. So, in the classroom, there is a limited number of us, and so there's not that much stuff going on in here, and so it's okay for us to just be in here, and this is one little compartment, okay? But, like 5 years ago, there was a problem in main hall on the other side here, like on the courtyard side, where all their windows started leaking over the summer. And when we came back to school at the beginning of the school year, there was like a ton of black mold in the classroom. It was like everywhere, okay? And it was way too late to say, hey, students, go someplace else. So what they did was they quarantined off that part of the building. They like sealed it all off with plastic, and they had like these, air conditioner blowers in that area. And then they took all those classrooms and they moved them to the gym, right? So for like 3 or 4 weeks, there were like 12 classes in the gym where they had all of the rows of desks set up, per class. And then the teacher would just stand in front of their section of desks and teach at their kids, right? Right next to another teacher. Now, thank God Rick Ferlita wasn't in there, because like you would have never heard anything else other than him. But it would still be very distracting to have to talk right next to another teacher and have to listen in a class where there was another class right next to you, right? Yes? Okay, so this is the point that I'm trying to make. In a prokaryotic cell, there's not that much going on at any given time, and so it's like being in a small classroom. But as the cells get bigger and bigger and bigger, there are all these conflicting things happening, and they can interfere with one another, okay? So, imagine we have a cell like this, a prokaryotic cell, and the prokaryotic cell is getting large, and it's going through some reactions, and those reactions are conflicting. You've got reaction 1 here and reaction 2 over here. And imagine that reaction 1 and reaction 2 conflict with each other, and they don't happen as well when they're right next to each other. Well, one of the things that we can observe in prokaryotes is prokaryotes will have an infolded area of their plasma membrane, okay? And that infolded area then has a sectioning effect where it says, okay, one thing can happen on one side of the infolded area and another thing can happen on another side of the infolded area. This is a way for them to separate reactions. Does that make sense? Okay. Well, over time, what happened was that these separations became totally infolded to the point where they were like that. Okay, so now these parts of the plasma membrane are actually touching on the outside. And so now you can have, if let's say reaction 2 is the one that's causing a major problem, you got reaction 1 over here and reaction 3, and those can be, you know, they can happen in the same area, but reaction 2 really causes the problem. You can confine reaction 2 into the inside of this totally compartmentalized area, right? And what that allows for it to happen is now, if it pinches off all the way then, you now just have a separate little space within the cell where reaction 2 can happen, and then reactions 1 and 3 can happen wherever they want within the cell. Make sense? So this is a prokaryotic cell over here. This is a infolded area. This is a compartmentalized cell. And this is an organelle. So the organelle really isn't anything special as much as it's just a sac of membrane where stuff can happen inside that doesn't interfere with the stuff that's outside. Does that make sense? Okay. There are a lot of organelles that were created in this way. In fact, every single organelle within the endomembrane system was created this way. So all organelles in the endomembrane system are the result of infolding. Okay, and one of the main pieces of evidence for this is, first of all, we can see prokaryotic cells infolded even today, right? We can even see compartmentalized prokaryotic cells, but there just aren't any prokaryotic cells that have organelles, so it's just one step off, right? The other thing that we can see is when we study their plasma membranes, if you look at a cell, so here's a big cell, okay? Let's say this cell has a red outer membrane and a blue inner membrane. Okay. Well, when we fold that membrane in, like this, whoops. Yeah, that's right. We're okay. When we fold the membrane in like this, and we now create a vesicle, or an infolded area, or a organelle, right? Now we've got the cell that has the outer plasma membrane that's red, the inner plasma membrane that's blue. But the organelles have exactly opposite that. The organelles have an outer plasma membrane that's blue and an inner plasma membrane that's red. And obviously they're not blue and red, but what we can tell is that the inner layer of the plasma membrane is unique from the outer membrane of the plasma membrane, because they're phospholipid bilayers, right? And so the bilayer has two layers, the inner side and the outer side. And so in a cell versus the organelle, the outer membrane of the cell will always be on the inner membrane of the organelle because of the way that they infolded. That makes sense? You guys get how that works?
So does that like prevent it getting out of it?
It doesn't really have any consequence as far as the way that the membrane functions. It just is a byproduct of how they were created. They were created by folding them inwards, and because you folded them inwards, the outer membrane becomes the inner membrane. So this is just like a piece of evidence that allows us to say, like, okay, this is what we thought happened, and then this is our confirmation of that. It also has a little bit of significance when we look at like the little name tags that are on the outside called glycoproteins and glycolipids, but we'll talk about that down the road here in a week or two. Okay? So, questions on infolding theory. All right. The second theory is called Endosymbiotic Theory. And endosymbiotic theory is not the part that explains the endomembrane system, but it's the theory that explains how we got our power-producing organelles. And essentially what it says is that a small prokaryote got eaten or engulfed by a larger prokaryote. Small prokaryote was engulfed by a larger prokaryote, which happens a lot. But the key here is that for whatever reason, in this particular instance in time, that small prokaryote didn't get digested. It just stayed inside of a vesicle within the large prokaryote. So the small prokaryote was useful and did not get digested. All right, so here's our large prokaryote, and here's our little guy, our small prokaryote. The large prokaryote engulfs the small prokaryote, like so. And then the large prokaryote then has this thing inside of it that contains the small prokaryote. And normally what would happen here is that it would pump in some hydrolytic enzymes, and those hydrolytic enzymes would break down that small prokaryote, and then it would digest it and eat it. But for whatever reason, in this instance, that didn't happen. And so this small prokaryote down here, let's say this small prokaryote was an expert at taking in glucose and making ATP, right? And so it just started exporting a bunch of ATP. Well, as it starts exporting a bunch of ATP, maybe there's some cellular process within this cell that prevents it from digesting it, right? Because it's being useful. And it's only going to digest things that are not useful. I don't know, something along those lines, right? And so for whatever reason, this thing doesn't get digested, and then it becomes an organelle in itself within this larger cell. Right? This seems far-fetched. So why would we have this theory if there wasn't evidence to back it up? Well, turns out there's a lot of evidence to back it up. Let's talk about some of it. First of all, this is how we got the endosymbiotic organelles that are mitochondria and chloroplasts. Okay, so we can say endosymbiotic organelles, which are mitochondria and chloroplasts. Contain double membranes. And you're like, big deal, who cares? Everything contains a phospholipid bilayer, and that's a double membrane, right? No, that's not what a double membrane is. A double membrane is 2 phospholipid bilayers, right? So it's got 4 layers of phospholipid, right? Now, that in itself is not that interesting. The thing that's interesting is that the outer layer is identical to the cell's membrane, and the inner layer is totally unique from anything else that we'd find in in that eukaryotic cell. So the outer membrane matches the rest of the cell. And the inner membrane is unique. Almost like it started out as a separate cell. Okay, well, if it started out as a separate cell, one of the things that we know should be there is that each of them should have their own DNA. Sure enough, endosymbiotic organelles have their own DNA that's separate from the host cell DNA. Now, I don't want to confuse you here. If you took mitochondria out of a cell and you like put them in a petri dish and you tried to culture them, they can't grow on their own. And this is because they've been together for billions of years, and a lot of the things that mitochondria would have needed to produce in order to live on its own were already being produced by the host cell. And so those parts of its genome got deleted over time. So it relies on the host cell to produce a lot of the things that they had in common, but it still produces its own unique proteins that were not being produced by the host cell. Okay? Other interesting things. Endosymbiotic organelles have their own ribosomes, and their ribosomes are totally unique from the ribosomes that you would find in a eukaryotic cell. Okay? Their own ribosomes. That resemble prokaryotic ribosomes. And the last piece of evidence is probably the least impressive piece of evidence, and that is just that they're the same size. So mitochondria and chloroplasts are roughly the same size as prokaryotic cells. Okay, now, this is all interesting, right? But here's the thing that I find mind-blowing, right? It is entirely likely, and when I say entirely likely, I mean like 99.9% true, that all of us, every single eukaryotic cell on the planet, so that's plants, animals, fungi, right? All of us, we all come from this one instance where one large prokaryotic cell engulfed one small prokaryotic cell. It happened one time, right? And then that gave it such an evolutionary benefit that that one eukaryotic cell then, that it engulfed this small one, then had a lineage where it divided, you know, quadrillions of times, quintillions of times, whatever the largest number you can think of as the number of divisions that it's gone through now. And it's changed a lot. Obviously, plants are different than animals. But we are the same in that we are both mitochondria-containing organelles or organisms. And so we know we had to come from this one instance. Okay? We know that mitochondria and chloroplasts both are the result of endosymbiotic theory, but we know that mitochondria had to come first because all eukaryotic cells have mitochondria. Plants have mitochondria. Animals have mitochondria. Fungi have mitochondria, but not all eukaryotic cells have chloroplasts. Plants have chloroplasts, we don't have chloroplasts, right? So that had to come second. Does that make sense? All right. Let's talk about the cells that are, the organelles that are part of the endomembrane system. Endomembrane organelles. So, let's talk about the endomembrane system, what it is, why it's unique. The important thing to realize about the endomembrane system is that they can share their membrane among different organelles. So, all organelles in the endomembrane system can share a membrane. And what I mean by this is when one organelle has to transfer something to another organelle, the way that they do that is they pinch off a little piece of their plasma membrane that's called a vesicle, and that vesicle travels to another organelle and then gets absorbed into it. So they actually transfer part of that organelle, part of that plasma membrane, to the other organelle. Okay, so we can say that vesicles bud off of a organelle or an organelle and attach to another organelle to transfer macromolecules. All right. Now, on the top of your reading guide, it says something to the effect of like, you need to not only know the names of the organelles and what they do, but you need to know the relationship between them. So that's kind of how we're going to go about this. We're going to follow something called the protein pathway. And this protein pathway is going to be specifically for proteins that are going to be exported from the cell. So proteins for export. Okay, so first thing, we've got the rough endoplasmic reticulum. So I don't have to write that anymore. I'm going to write ER. I'm going to call it the ER, the rough ER. Okay, so the rough ER's job is to hold on to these things. They're called bound ribosomes. Bound ribosomes are a type of ribosome that attaches to the rough ER. Okay, so we can say that it's a large sac of membrane that contains pores where ribosomes attach. Okay? We've talked about this before, but ribosomes are the things that make proteins, and therefore we can say that the rough endoplasmic reticulum's job is to make proteins, but it's going to make proteins using ribosomes. Okay? So we can say that proteins are produced inside the rough ER by bound ribosomes. We'll do a little drawing over here. So blue is going to represent plasma membrane. So here's my rough ER. Mostly phospholipid. And because ribosomes are made out of RNA mostly, we're going to do them in green. You can see how this thing is studded with these green things here. And that's why we call it the rough ER, because under a microscope it looks rough because it's got a whole bunch of ribosomes on it. And let's say that this ribosome on the very tip here creates a protein. So now there's a protein inside of this ER. That's the red thing is at the end there. That's a protein. Okay? So, in order for this protein to go anywhere, the ER needs to create a vesicle. And it's going to do that by budding off a piece of its membrane that contains that protein. This piece of membrane that buds off is called a vesicle. So when it's all budded off and it contains the protein, we refer to it as a vesicle. Vesicle is a small sac of membrane used to transport macromolecules from one organelle to another.
What's it say after small?
Sac. Small sac of membrane used to transport macromolecules from one organelle to another. All right, so question is, then how does that happen? So in bio honors, you kind of just skip over this point. You're like, it floats around the cell and then gets to where it needs to go. It doesn't really do that at all. It actually is transported purposely to a specific area, and it goes along an area that's called the cytoskeleton. And I'm going to indent cytoskeleton here because cytoskeleton is not part of the endomembrane system on itself because it doesn't have a membrane at all, but it's required for this protein pathway. Okay, so we can say cytoskeleton provides a path for the vesicle to travel on. All right, so then here's our cytoskeleton. Let's say it's got like a couple branches or something like that. A couple different places it can go. And so this vesicle will actually be attached to a motor protein. Okay, and a motor protein will then walk it down a certain path on the cytoskeleton here until it gets to where it needs to go. Okay, let's say that this one happens to be going to an area that's called the Golgi Bodies. The Golgi Bodies used to be called the Golgi Apparatus, but they changed the name to the Golgi Bodies to try to get across the point that they are not always interconnected, right? So you can see that this is a Golgi body, this is a Golgi body, this is a Golgi body. They're not hooked up to each other, okay? So the Golgi Bodies are sacs of membrane that help proteins fold into their final form. So you know how I said that a protein would fold differently if it was in a hydrophobic environment versus a hydrophilic environment? Well, maybe the inside of one of these Golgi bodies over here is a hydrophobic environment. So when you send the protein into it, it changes the way that it's folded up into its final form. Okay? So now you've got the protein that enters into here. Okay? And then it's going to travel through here, like so. And let's say by the time it gets to the end, now it is in a different shape. Now it's in this upside down U shape or something. It's refolded itself. Then we're going to have another vesicle butt off. See, another vesicle forms. And then it's going to go to its final destination, which is probably going to be the cell membrane. Cell membrane is the membrane that separates the outside of the cell from the inside. Outside environment from the inside. Okay? So, how does it do that? Here's the cell membrane. It's actually going to get absorbed by the cell membrane, and this protein will then be exported without a vesicle. It will just go out and be exported from the cell. And that process is called exocytosis. Yeah.
So does that happen like every time it enters a new organelle, like from?
Yes, this part. Yeah. So this budding off and then absorbing anytime it transfers between an organelle. That's what it does. Yep. And so that's a good point. That means that the rough ER is constantly getting smaller, and the Golgi apparatus is constantly getting bigger, right? Because the rough ER keeps budding off these vesicles, and then they get absorbed by the Golgi apparatus. But then the Golgi apparatus then buds them off in itself, and then they end up in the cell membrane. So the cell membrane is constantly getting bigger, right? Well, the other thing the cell membrane is doing is taking in things through a process called endocytosis, and so it's creating vesicles on the inside. But the thing that is happening is that the rough ER is getting smaller and smaller all the time, okay? So that's a good segue into the next thing here, which is the lipid pathway. Actually, before we do that, let's draw some arrows. Okay, so this is the cell membrane here. This is the Golgi bodies there. This is the cytoskeleton here. This is the vesicle here. And this is the rough ER there. Good? Okay. Lipid pathway is much shorter. So the lipid pathway only involves one organelle, and that is the smooth ER. And the smooth ER's job is to produce plasma membrane and also to produce lipids. So a large sac of membrane that produces more membrane, more plasma membrane, and other lipids. But it's mostly producing phospholipids, right? And so that means that anytime something needs to grow in a cell, the way that it grows is by getting more plasma membrane. And so you can't get more plasma membrane unless you are receiving that plasma membrane from the smooth ER. So the smooth ER's main job is to just make vesicles that contain nothing, right? And send empty vesicles out to organelles that need to grow. Sends empty vesicles to organelles that need to grow. So, for instance, if we were saying, like, in this stage of the cell cycle, the cell grows, well, what we're talking about is the smooth ER is sending the cell membrane a whole bunch of empty vesicles that allows for the cell membrane to get bigger, right? Okay. Next, let's talk about digestion and detoxification. Lysosomes are sacs of membrane that contain digestive enzymes, so small sacs of membrane. That contain digestive enzymes or hydrolytic enzymes. What process do you think hydrolytic enzymes are involved in? Hydrolysis, right? And hydrolysis reactions are reactions where large molecules get broken into small ones. Okay, so they break large molecules into smaller ones, which we refer to as intracellular digestion. Intra means within, so anytime the cell needs to digest something, it's going to use a lysosome to do that, intracellular digestion. Okay. Second thing is called a peroxisome. And peroxisomes, their main job is to detoxify. They contain enzymes that detoxify the cell. So you guys are all exposed to toxins on a regular basis, right? Some of them are even produced by your body. Urea is a good example of a toxin that's produced by your body. It's produced through protein catabolism. But peroxisomes are there to sort of help you out. And so your liver and your kidneys contain tons of these things called peroxisomes. They're organelles, or the cells in your liver and the cells in your kidneys contain these things called peroxisomes. And anytime that you're exposed to a toxin, they go and they oxidize it and they create hydrogen peroxide as a byproduct, and then your body breaks down the hydrogen peroxide. So essentially every time that a peroxisome interacts with the molecule, it becomes less and less toxic. Sometimes it takes a couple of go-rounds for it to finally get to a point where it's not toxic at all. But it's always going from something that's very toxic to something that is less toxic. Okay. Last class here are the storage organelles. Storage organelles contain the vacuoles. Okay, vacuoles. Vacuoles. Vacuoles are long-term storage, so large long-term storage. Large sacs of membrane. For storing food and water. There's two main types of these. The first type is called a food vacuole. You could probably guess what a food vacuole holds. Food, right? And then the other type are the ones that hold water. Okay, so there's two main ones that hold water. One of them is called a contractile vacuole, and its entire job is to actually get water out of the cell. So in freshwater organisms, water is always trying to diffuse in through osmosis. And if you allowed that to happen, then they would eventually just explode because they would constantly have water going in. Well, the contractile vacuole takes in a bunch of water and then squeezes to squirt the water back out. So the water is constantly going in through osmosis, and then the contractile vacuole is constantly squeezing it back out. Right? That's in contrast to in plants that have this thing called the central vacuole. And the central vacuole's main job is to fill up with water, and it fills up with water in order to stretch out the cell. Okay, so anytime that a plant cell grows, it grows by filling up its central vacuole with water and stretching out the cell so that it touches the cell walls. The central vacuole is huge. It's the largest organelle. It's only in plants. But the central vacuole can take up to 90% of the interior volume of a plant cell, right? Just one big water-filled vacuole. Okay. The last thing in the storage is the nucleus, which is the nuclear envelope. And then the nucleolus, but the nuclear envelope is the part that we are concerned with right now. And the nuclear envelope's main job is to be a large sac of membrane that has pores in it. Large sac of membrane that contains pores. Pores are, of course, small holes. And those pores only allow certain things to go in and certain things to come out. Okay, so the main job of the nucleus is, of course, to protect the DNA. So it protects and stores DNA. So, as I said, it is vastly important that you know all of these organelles, right? You have to, that's like step one. You got to know this organelle does this, and then the relationships between them is step two. So that's really important for the AP exam. It's going to be really important for the Unit 2 test. So we're going to continue to sort of harp on this relationship between the organelles. Does anybody have any questions?